Well, welcome back to another Monday. It looks like we're a little bit thinner today than we normally are. I don't know if that's good or bad. Maybe I scared some people off. I didn't mean to. We have an exam next week on Wednesday. Okay. It's in the first introductory slideshow. All those exam dates are indicated, so make sure you be aware of those. We have one next Wednesday. It will cover up through chapter 4. Right? What? Yes. The exams, you'll recall, are all multiple choice and true-false, which is the worst way to assess what people know, but unfortunately I don't have time to grade 160 essay exams. It's just physically impossible. Yeah, I'm sure you would. You know, at Davis, I routinely had classes that had 200 people. My upper division genetics had 200 people in it. And we just got used to it. But we had an army of TAs. I had a half a dozen TAs to help grade. So I don't have that situation here. So unfortunately, they are multiple choice and true-false. That goes for the folks at home as well. Those of you who are DDE students should be aware that once the lecture for Friday, that'll be 2 lectures from now, once that's finished, you should be gearing up to take that exam as well. Okay. And remember that Monday next week will be a review. I'll have a practice exam for us to go through so you get some idea of the kinds of questions that are asked. But be aware that it does cover the material up through chapter 4, and it includes both lecture and book. Okay, you can't not crack the book and get by in this class. You've got to read it. Part of the reason is because about half to 3/4 of the questions will be coming from a test bank, and test banks, as a general rule, emphasize what's in the book. Now, a lot of the stuff that's in the book I've covered, but there are some things I haven't. Using radioisotopes for medical purposes, for example, I didn't cover much, and that's in the book. So that's something you should be aware of. Okay, so we're trying to finish up chapter 3 and move into chapter 4 this week, and we've looked at 2 of the 4 categories of macromolecules on Friday. We looked at polysaccharides and we looked at lipids, and what I want to do is just continue with chapter 3 and move on to proteins, which are another major category of macromolecule in the cell. They're made up of chains of amino acids. That's the monomer unit. Okay? The name amino acid, and I'll show you a figure of one, refers to the fact that they have an amino group on one end and a carboxylic acid group on the other end. That's why they're called amino acids. Okay, so there's an amino group on one end, a carboxyl on the other. So they tend to be charged. The carboxyl tends to drop off a hydrogen and therefore become negatively charged at cellular pH, but then the amino group picks up a hydrogen. So there's no net change in pH typically. And there's only 20 amino acids that are used by all living organisms. There are some funny variations that living organisms will do to those, but it's after the protein is assembled. Sometimes they'll put other functional groups on those amino acids, like a hydroxyl group or something like that. Okay. This is an amino acid. Okay. They're easy to recognize because they always have this same kind of structure. They have a central carbon atom, okay, right up there in red. They have an amino group on the left side of this one and a carboxyl group, or carboxylic acid group, on the other. One of the bonds of the central carbon is filled up with hydrogen, and then the 4th bond of that central carbon is filled up by something we call an R group. That R in organic chemistry simply indicates an additional structure that varies according to which amino acid you're looking at. All right? So that's a side chain that's specific to the amino acid being examined. So with 20 different amino acids, there's 20 different R groups we can look at, and those R groups have different properties. Okay, so here's some examples of amino acids. There are a number of different categories that we can fit these into. Leucine, for example, is hydrophobic. Here's the amino acid part of this thing. And then here's the R group, and it's just a branched hydrocarbon. And as you can imagine, that's very hydrophobic. It's not polar. It's just carbon and hydrogens. There are others that are hydrophilic. Serine, for example, has a hydroxyl group on it. So it's clearly a hydrophilic amino acid with that hydroxyl. Cysteine has a sulfhydryl group as part of its R group, and so it tends to be hydrophilic as well. Okay? This cysteine is really nice because that sulfur can form bonds with other sulfurs and other cysteines, and that helps to hold proteins together, as we'll see. All right, so there's an example of 3 of them. We won't go into a lot of detail on the side chains, but do be aware that the side chains are what really give the protein its characteristic shape and its characteristic functions. All right, you shouldn't be surprised that these proteins are assembled by dehydration reactions. All right? All of the molecules, macromolecules we've looked at so far, are assembled by dehydration reactions. So amino acids, polypeptides are no exception. All right, simple reaction. You take the carboxyl, as shown on the left of this figure. You take the carboxyl of one end, the OH off, the hydrogen of one of the amino groups off of the other amino acid, and with that dehydration synthesis, you form a bond between the 2 amino acids, right there between that carbon of the carboxylic acid and the nitrogen of the amino group. Okay, we call that a peptide bond, but it's no different than any other covalent bond, right? It's just a covalent bond. Saying it's a peptide bond simply indicates that it's a bond between amino acids in a polypeptide. Okay, so it's no different than any other covalent bond. One thing I mentioned last time, and I want to emphasize it again, is I tend to use polypeptide and protein differently. Your book doesn't, and it should. A polypeptide may in fact be a functional protein, but it isn't always, because some proteins will have several polypeptides that contribute to that protein. A polypeptide is just a linear sequence of amino acids that are all tied together by peptide bonds or covalent bonds. All right? You can fold those up, roll them up into specific shapes, and then you can take 2 of those and stick them together. All right? And it's no longer a polypeptide. There's 2 polypeptides making up that protein. All right? So many proteins have several units, several subunits to them. Each of those subunits is a separate polypeptide. So that's why I use the terms differently, and I'll show you that as we get into the 3-dimensional structure of proteins. Okay. Proteins don't exist as linear chains of amino acids in living cells. Okay? They fold. They form these funny different conformations or shapes to the polypeptide. And the shapes that they form dictate the function that that polypeptide chain is going to have. So for example, this particular polypeptide chain that we're just showing the backbone of the polypeptide as a ribbon. It forms a whole bunch of helical structures and sheets. So here's a helix that has formed in that backbone. Here's another helix, another helix. There's a couple of sheets up here where we've got side chains interacting as well as the backbone of the amino acids interacting. So they form a lot of 3-dimensional structure. So when you're describing and trying to figure out what proteins do, you have to be aware of the 3-dimensional structure. That's not easy to determine. There are some techniques to do that, but they're sort of beyond the scope of this class. Suffice it to say that we can use techniques to determine 3-dimensional structure of proteins and figure out what's where. Okay, so they form lots of additional structure, and that results in the correct or appropriate function for the protein. In fact, there's a whole set of proteins in cells that are used to fold proteins correctly. Right? There's a whole machinery there to make sure the proteins are folded the way they're supposed to be folded. If they don't fold correctly, then the cell gets rid of them. It kicks them out, discards them, and then it starts over. Okay. All right, so protein structure is described by 4 different levels. Not surprisingly, they're called primary, secondary, tertiary, and quaternary. First, second, third, and fourth level. All right. The first level of protein structure is the primary level, and that's just the linear sequence of amino acids. What we're doing is using a ribbon to simply represent the amino acids that are present, and if we simply rattled off which amino acids were present in what sequence, that would be the primary structure. A lot of times that's the first thing that we're interested in. We might want to be able to determine the sequence of this protein so that we could find out some more about its function. Right? What is it about that sequence of amino acids that leads to the function of the protein? Okay? But you don't find this sort of primary structure in living cells. When the protein has been assembled, you find lots of other levels of structure. So we go to the next level, and that's the secondary structure. Okay, as is shown up here in the second part of this figure. There's 2 different kinds of things you can form a secondary structure. The alpha helix and the beta sheet. So in that protein I showed you before, those curlicue or sort of spiral staircase structures, those were alpha helices. They form because you get hydrogen bonds between oxygens and nitrogens in the backbone of the polypeptide. Okay, it all forms because of hydrogen bonding. And that forces the amino acids to adopt a helical shape. Okay? In other cases, you form what's called a pleated sheet. It's pleated because of the bond angles here. The carbon and nitrogen bond angles cause it to be pleated. But what you've got here basically is a line of amino acids interacting with another part of the amino acid sequence, again by hydrogen bonds. Here's an oxygen associating with a nitrogen-hydrogen pair. And here's a nitrogen-hydrogen associating with a carbon-oxygen on the other side. Okay? So really all you need to remember about these secondary structures is that you form helices or sheets, and they're formed by hydrogen bonding in the backbone of the amino acid sequence. Okay? That's what you need to know. All of these amino acids have side chains, and those side chains interact as well. Hydrophobic side chains tend to clump together because they're excluding water. Okay? Hydrophilic side chains tend to clump together because they interact with water. Charged amino acids tend to interact with each other if the charges are opposite. In a sense, they form ionic bonds. Okay? So the side chains interact as well, and what that can do is that can take helices, for example, and fold them around so they interact with each other. Or you can take sheets and twist them so that they form a different kind of structure. So that side chain interaction is called the tertiary structure of the polypeptide. The tertiary structure refers to all of the side chain interactions that cause all of these alpha helices, there's one right here, and these pleated sheets, to fold up on themselves to create what we call a globular protein, a nice 3-dimensional structure for this polypeptide. Okay, so that leads to the 3-dimensional shape of this polypeptide, all of those interactions among side chains. Now, by itself, this polypeptide may be functional, but in many cases, proteins actually have many polypeptides contributing to the final functional form. That's what's happening here, and we refer to that as quaternary structure. If you have multiple subunits, multiple polypeptides contributing to the final protein, then you have quaternary structure. So this protein, transthyretin, has 4 identical polypeptides contributing to its structure. Okay, they're all sticking together. Why? Well, it could be covalent bonds forming between the sulfurs in those cysteine amino acids. Could be ionic bonds that are forming, lots of hydrogen bonds that are forming between them. Whatever the bond, or bonds that are forming, you get multiple subunits contributing to the protein, and that's quaternary structure. All right, so primary, secondary, tertiary, and quaternary structure for proteins. Okay, any questions on proteins before we go on? That's their basic structure. Okay, I'm not going to say a lot about nucleic acids because when we get into molecular genetics next term, we'll talk a lot more about the structure and function of nucleic acids. They are the genetic material of the cell. There are basically two kinds of nucleic acids, DNA and RNA. Right? And depending on the type of organism you're looking at, actually RNA may be the genetic material, but for most organisms, DNA is the genetic material. Okay, so what are nucleic acids made of? They're made of nucleotides. That's the monomer. All right, so in all of these macromolecules we've looked at, we find that they are simply made up of many similar subunits. The polysaccharides are made up of simple sugars. Proteins are made up of amino acids. Nucleic acids are made up of nucleotides. Okay, so two major types of nucleic acids known, DNA and RNA. Thankfully, you don't have to refer to the name deoxyribonucleic acid. You can just say DNA and everybody knows what you mean. Okay? They serve as storage and transport forms of the genetic material. Okay? So in most organisms, DNA is the storage form of the genetic material. It's your archive. That's the library. Okay? And the RNA in the cell is a transport form. It's as if you're taking a book off the shelf in the library, you make a Xerox of it, you walk out of the library with a Xerox. That Xerox is the RNA. That's the form that the cell uses and disposes of when it's done. Okay? It would not be good for the cell to dispose of its archival copy, so it keeps that separated and sequestered in the nucleus if we're talking about a eukaryote. Right, so it just keeps that separate. It reads it, makes a copy of it, and then uses that copy as the usable form. Okay, this is a nucleotide. It's got several distinctive structures that clearly identify it as a nucleotide. Right, so three components. The first component is a 5-carbon sugar. Right, you see that right in the middle here in this sort of bluish lavender sort of color. You know that's a sugar because it's a ring with oxygen in the ring. And that's how you identify sugars if you're just looking at them. And it's got lots of hydroxyls as well. There's an OH down there that you can easily see. Okay, so 5-carbon sugar. I just indicated that it's either ribose or deoxyribose. You don't have to worry about the distinction between the two for right now. We'll get to that much later on. The only difference between the two is how many hydroxyl groups they've got. This one is deoxyribose. A phosphate group, okay, that's shown on the left in yellow. And then a nitrogen-containing base, or a nitrogenous base. Nitrogenous just means it contains nitrogen. Okay, so you notice that these rings in this molecule have nitrogen in the rings. Okay, this one happens to be the base adenine. I know that because it tells me so, but there's a amino group up here. Okay, so I know that's adenine. There's 4 used in DNA: adenine, guanine, cytosine, and thymine, which we just abbreviate A, G, C, and T. Okay? The sequence of those bases is the actual information content in DNA. It's that simple. Okay? The various sequences of bases mean something. In fact, they are the instructions for making polypeptides, proteins. Okay, so that's really all that's involved in the genetic material. It's a blueprint for making proteins. But boy, what those proteins do makes all the difference in the world. All right, so how do you assemble these things together? Well, what you do is you take the hydroxyl of one of them and you stick it to the phosphate of the next by a dehydration reaction. No surprise there. Okay, so if we brought in another one of these nucleotides, the phosphate would be right down here. We'd just take that phosphate, stick it to this hydroxyl, and we'd have two nucleotides stuck together. All right, that bond is called a phosphodiester bond, which, you know, it indicates that it's that sort of covalent bond in DNA, but we don't really have to worry about whether it's anything special. It's just a covalent bond. Okay, so what we end up getting with nucleic acids is a double-stranded molecule. Right? We call it a double helix. The reason this happens is because if you take a long stretch of nucleotides and string them all together, right, those will interact with other nucleotides on another strand by very specific rules. Okay, the strands are complementary to each other. They're not identical. Right? They're held together by hydrogen bonding between bases. Okay? It's always A with T, adenine with thymine. It's always cytosine with guanine. Okay? That's always the base pairing. Okay? A with T, G with C. If you don't see A's with T's and G's with C's, then you've got a very, very, very unusual case, or you're looking at a mutation, an alteration in the sequence of the bases. It's really all a mutation is. And there's a few extremely rare exceptions I can think of. Long stretches of G's will pair with each other to form triple helices, where you have three strands together. Really, really unusual. We don't even know if that forms in living organisms. Okay? But we can get it to form in a laboratory setting. All right, so always A's with T's, G's with C's. And those hydrogen bonds, when you add up all of the strength of those fairly weak hydrogen bonds, you've got a nice strong molecule that holds together really well. Okay, so we won't deal much more with nucleic acids except to give you the basic structure, because, as I said, when we get into molecular genetics, we're going to go into this in a lot greater detail. You'll be sick of understanding what the sequence of bases really means. Okay, so there's a whirlwind tour of biological molecules in the cell. And what I want to do then is just move right on into Chapter 4. Oops. Okay, which deals with cellular components. This is a chapter that's particularly difficult for me to do because I teach at the 400 level, the senior level. I teach cell structure and function, which is basically a semester-long survey of how cells do what they do at the molecular level, except it's crunched into 10-week terms. And then I also teach an advanced cell biology course that's even higher level than that. So it's really difficult for me to take all of the knowledge that I have about the cell and all of the stuff that cells do and crunch it into a tiny little synopsis. I'll try my best. All right, but if I get off on tangents, please forgive me, because this stuff is it's way cool. What a lot of people come out of this class seeing is how intricate things are at the cellular and molecular level. I've had so many people walk away saying, God, I had no idea that cells were so jam-packed with stuff, doing all these different functions. So it's kind of fun to just get the wow factor there. But what my purpose is in this chapter is to basically survey what the major cell components are. What do cells do? What do they have in them that do these different functions? And what we'll do over the rest of the term basically is examine those functions for a few selected organelles in the cell. Okay, so the cell is the smallest fundamental unit of life. I've said that several times. If you break open a cell, you have individual components that can do some of the functions of living organisms, but it's not alive. It doesn't do all of them. Right? So the cell in biology is analogous to the atom in chemistry or physics. It's the smallest fundamental unit of matter in chemistry or physics. Well, the cell is the smallest unit of life in biology. Okay? Cell architecture varies according to function and according to whether it's prokaryotic or eukaryotic. Eukaryotes tend to be bigger than probes. All right? But there is an upper limit to size. And it has to do with a little thing we call the surface area to volume problem, which I'll describe, and your book describes that as well. Okay? Cells can be prokaryotic or eukaryotic. There are fundamental differences between prokaryotic cells and eukaryotic cells. Right? Fundamental differences, which I'll go through. Cells contain these little structures called organelles. And those organelles have individual functions in the cell. The chloroplast, for example, provides the photosynthesis of the cell, the ability to capture light energy and make sugars from that light energy. The mitochondrion provides energy conversion processes for the cell. The nucleus provides a protective mechanism for the genetic material. All right, so all of these organelles provide us a very specific function. Okay, if you are multicellular, and even to some extent if you are one-celled, you must be able to communicate with other cells. If you're multicellular, you've got to be able to connect with other cells to form tissues. All right, strong tissues that can resist tearing. All right, so that's a function of some of the cell organelles that we'll look at. Okay, how do we look at cells? Cells are small structures. Most of them are beyond our ability to see with the naked eye. So we have to use microscopes. Those are a key tool. Not just light microscopes. Okay? Also, we use electron microscopes, which can see things that light microscopes simply cannot resolve. All right, so light microscope, the scanning electron microscope, which scans the outside of a structure, and then the transmission electron microscope, which actually sort of transmits electrons through the structure so we can view internal structure. Fluorescence microscopy. Fluorescence microscopy takes advantage of the fact that some molecules will absorb light. Okay? Chlorophyll, for example, is a molecule that absorbs light. It then emits that extra energy as light as well. Okay? So it absorbs light and then emits light, and that emission of light is called fluorescence. Right? So what we can do is we can use molecules that absorb light and emit it back as tags in the cell to label or to look at specific cell components. Okay? Biochemical and cell biology techniques are used as well. If you're real curious about those, we won't talk about most of those in this class. They're sort of beyond the scope. But if you are interested, you should take cell biology, because we talk about those quite a bit. So there's lots of biochemical and cell biology techniques that we can use. All right, this is a light microscope. Probably most of you are familiar with light microscopes. You may have used them in previous classes or in previous schooling. They're actually a fairly straightforward structure. You've got a light source of some type, which passes light through what's called a condenser lens. And that condenser lens simply focuses all of that light onto a small area, the specimen, whatever you're trying to look at. Okay? That light then passes through an objective lens, which magnifies the object. So there's the objective lenses up there. And each of those has a different magnification depending on the size and shape of the lens. Okay, so the object is magnified. The light then passes through the ocular lens, which is the lens that you're actually looking through. And that usually magnifies it some more, typically 10x or 20x, 10 times or 20 times. Okay, and then you're able to actually view that image. So you get a fair amount of magnification out of this setup. The resolution depends on what wavelength of light you use. Resolution refers to what's the smallest object you can see using that wavelength of light. Okay. The resolving power of a microscope is a different thing, though. The resolving power is the ability to distinguish objects as being separate objects. Okay, so if you have low resolving power, you may see two things that are together, but they'll look like they're one object. If you have better resolving power, then those resolve into two separate objects. Okay, so it's the wavelength of light that matters. The shorter the wavelength, the better resolving power. That's why blue light is much better to use. Blue or violet light is much better to use than red light. Okay, so light microscopes are fairly straightforward. This monstrous thing is a transmission electron microscope. The actual microscope is this orange tube right here. Okay. It's sealed, and inside there would be a vacuum that's created. The reason is because you're not using visible light now, you're using a stream of electrons. Okay, so you generate a stream of electrons, and they bump or bounce off of your object, and then they bounce onto a screen where you can actually detect them. And that's what you see. You have to keep a vacuum in here, because if you didn't, then those electrons would bounce off of stuff in the air in the tube, and you wouldn't be able to get good resolution. Okay. So what you see then is the actual image on a computer screen. You can't actually look at your object directly. Okay. The material has to be preserved and dehydrated, because you can't put living material in. It's a vacuum, after all. And you have to have for a transmission electron microscope, you have to have extremely thin material. Okay? The material needs to be a micrometer or less in thickness. Remember, a micrometer is 1/100,000th of a meter. Very tiny. Okay? But the resolving power of these things is tremendous. You can see large, with some electron microscopes, you can see individual atoms on sheets of material. So it's pretty amazing. Here's a scanning electron micrograph of some cilia on the surface of a cell. These are little hair-like projections off the surface of the cell. The scanning electron microscope is particularly useful for looking at external features. What you do is you take your specimen, you coat it with a heavy metal. Gold or platinum are the metals of choice. Okay, it's sort of fun to go into these electron microscopy labs, because they have these electroplating machines, and the whole inside of it, the machine has been electroplated with gold, really thin layer of gold. They're just beautiful things. So electron microscopy is somewhat expensive, because you have to use gold or platinum as your metal. But what happens then is the electrons bounce off of that metal that's coating your specimen, and that's what you see. All right, so you're not seeing the organic material here, you're seeing the electrons bouncing off of the metal that these have been coated with. But it's unparalleled for looking at external surface features. All right, it's really good. No live material allowed, obviously. Okay, fluorescence microscopy. The book doesn't talk a lot about fluorescence microscopy. Okay, they're extremely useful for studying cells. And as I said, some molecules will fluoresce. They absorb light, and then they spit it out at a different wavelength. So a molecule might absorb, say, blue light and spit out green light. Right? Well, you can detect that green light. The neat thing is that you can use these molecules to tag specific cellular structures. You want to tag the nucleus? Easily done. You just use a fluorescent marker that binds to the nucleus. DNA can be tagged really easily. There are several dyes that are fluorescent, and you can use those for DNA. DAPI is one. Hooks 33258 is one. There's several of those. They all fluoresce blue. So you can look at the DNA in the nucleus of the cell. Okay, so it's a really neat technique. And here's an example of this technique. This cell has been multiply labeled with different fluorescent dyes. Okay. We're seeing DNA labeled pink. It's a little bit difficult to see because you've got that blue streak in there where you're getting interaction between the pink and the green. All right, but if you look carefully, you can see little pink lines in the center of this cell. That's DNA. Okay? The cell is getting ready to divide, and so the DNA is condensed, it's coalesced to the chromosomes. The structures separating the DNA between the cells, these are microtubules, and those have been labeled green. All right, so you can label cells with multiple fluorescent tags, and A, you get really pretty pictures, and B, you can see what's going on in the living cell. Many of these are what we call vital stains or vital dyes. It means we can stain the cell or treat the cell with these things, and it doesn't kill it. So we can watch how the cell changes over time. All right? We can watch these microtubules and ask the question, do they get bigger, do they get smaller? How fast does that happen? How often does that happen? Okay? We can learn so much more about the cell because it's the living cell that we're observing rather than a dead representation in an electron microscope. Okay? So multiple labels can be used. Okay, so you want to study the external shape of the small hairs on the antenna of a fly. The best method to use would be what? Okay. The key to answering this question is to look at what the researcher is trying to do specifically. And there's a key phrase here, external shape. Okay, that's the key phrase. You're also looking at these small hairs on the antenna of a fly, which are pretty darn small. Yeah, you could use a light microscope to do that, but your resolving power isn't going to be very good. All right, so light microscope is probably not going to be the best answer. For that reason, fluorescence microscopy is not going to be the best answer either. And the reason is because fluorescence is just light microscopy. It just happens that you're looking at specific types of light. All right, so you're left with transmission electron microscopy or scanning electron microscopy. Transmission is passage of electrons through a very thin section of your sample. That's not good for external structure. The best thing for external structure, to study details of external structure, is scanning electron microscopy. So for that reason, the answer is D. It allows detailed observation of external structures. Okay. You could argue that light microscopy is an appropriate answer, but the question says the best. And while you could use electron or light microscopy, it's not as good as SEM. All right. One thing we need to be aware of before we actually start looking at the structures of cells and how big cells are is a sense of size. All right? There aren't very many single cells you can resolve with the naked eye. Our eye can resolve things about to 0.2 of a millimeter at the very smallest. That's some of the larger of the cells. Certain protists might be that size. Okay? So most of the time you're going to have to use microscopy. Okay, so how small is small? You need to have a sort of perspective on what we're looking at here. So books always give you this kind of perspective. All right, there's human height, perhaps around 1.5 meters, 2 meters on average. Okay, slightly less than 2 meters. That's actually the length of some nerve cells. Nerve cells that run from, say, your spinal column to the tip of your toe, that's one cell. All right, so that's a meter in length. Okay, that's pretty long for a cell, but still the diameter is very small. You wouldn't be able to see that cell except with a microscope. All right, a chicken egg is a single cell before it's fertilized. All right, so that's pretty big. Frog eggs get down around a millimeter or 2 in size. Okay, they're 1/1000th the height of a human. Okay, so the smallest object we can discern, as I said, is about 0.2 of a millimeter, and that's for a good eye. Beyond that, you're going to need a microscope because everything else is smaller. And basically, that's the realm of the cell. So we look at the bottom half of this figure, and we're looking at things we can resolve with, say, the light microscope. Okay, most plant and animal cells have an upper limit of about 100 micrometers. 1 micrometer is 1 × 10⁻⁶ meters. Okay, bacteria are even smaller, down to about 1 micrometer. Some of the smallest bacterial type cells, these mycoplasmas, are very, very small, maybe 200 nanometers. A nanometer is 1 × 10⁻⁹ meters. Right? Extremely small. Probably most of you have actually had experience with mycoplasmas. There's one species that causes tuberculosis, Mycobacterium tuberculum. There is another species that causes leprosy, Mycobacterium leprae. There is a third species, Mycobacterium genitalium, which almost everyone has living in the cells in their reproductive tract. Okay, they're just freeloaders, basically. Can't get rid of them. Right? Sometimes they get in the wrong places and they cause us trouble. Other than that, no big deal. Okay, and they're some of the smallest cells that you can think about. All right, 1 nanometer, you're getting down to the size of viruses. Viruses cannot be resolved except by the electron microscope. You can't see viruses in the light microscope. They're just too darn small. You must use an electron microscope for those. Viruses are getting close to the size of some of the organelles in the cell, like ribosomes, which are involved in making proteins. Okay, we get to smaller sizes, proteins 10 nanometers to about 5 nanometers. Lipids are even smaller, down to about 1 nanometer. Some of the small molecules, like water is being shown here, smaller than that, so 0.5 nanometer in diameter. All right, really small. We get down to the size of atoms, and we're dealing not in nanometers anymore, but what we call angstroms. Okay? An angstrom is 1 × 10⁻⁻⁰ meters. All right, so extremely small. So this sort of gives you a perspective on what we're dealing with when we're dealing with cells and their subcellular structures. Very, very small things. Okay, so in case you're not familiar with the metric system, you're going to want to use this table. Hopefully most of you are, but we here in America have not followed the rest of the world and switched to the metric system. It makes a lot more sense, but we're sort of stuck with our imperial measurements, which we continue to use. Okay, so a centimeter is 10⁻⁻² of a meter. The basic unit of length is a meter. The basic unit of volume is a liter. Okay, so meters and liters. The prefixes denote powers of 10 relative to the base measurement. So you're talking about a kilometer. Kilometer is 1,000 meters, kilometer. A kilogram, it's a unit of weight, right? 1,000 grams. For liters, a kiloliter is 1,000 liter. The kilo means the same thing. It means times 10 to the third relative to the base measurement. Okay? So most of the time we're dealing with units of length, so we deal with meters. All right, so micrometer, nanometer I've used already. All right, so 1 meter is 100 centimeters, 1,000 millimeters, 10 to the 6 micrometers, and 10 to the 9th nanometers. Okay. There is a problem that cells face, and that is that surface area and volume don't increase equivalently. All right, when you're calculating surface area, you're calculating basically length times width. Okay. You've got 2 dimensions there. When you're calculating volume, you've got 3 dimensions. So surface area varies as the square, but volume varies as the cube. All right, so as a cell increases in surface area, its volume increases even more, to the point where the surface area simply cannot supply the volume of the cell. All right, the cell has too much volume for the surface area to be able to allow for gas exchange, obtaining nutrients, all those sorts of things. This figure illustrates that. If we have one large cube that's 30 micrometers on a side, then its surface area is 5,400 micrometers, square micrometers. Right? What if we break that cube up into littler cubes that are 10 micrometers on a side? All right, the surface area now is 16,200 square micrometers. And that's much more surface area. It's much easier for the total volume of those cells to be served by that surface area. So there's an upper limit to the size of cells, and it's because the surface area varies as the square, but volume varies as the cube. Okay? So there's no such thing as giant amoebas eating Detroit, not to mention the blob, whatever it happened to be. Right, do you ever see that movie, classic, Steve McQueen? I can't believe Steve McQueen would actually play in that classic version of the blob. He must have been desperate for money. They made a remake of it recently, too. I haven't watched it. The original was enough. Right? So, size constraint on cells. We'll stop there and continue at this point on Wednesday.